Okay, hello everyone, welcome to this demonstration of required practical 4 about the leakage of beetroot pigment from slices of beetroot depending on the amount of ethanol. So, I'm just going to run you very quickly through what we need to do as far as the method goes. You can see the method on the side there. I'm going to start with the first part of making up the colour standards, as you can see on the method, right, which is on the sheet just here. Okay, so first part of the method, going through this exactly as you would were you doing this practical yourself. Use the beetroot extract and water to prepare a series of 6 test tubes containing 5 cm cubed of different proportions of beetroot extract and water. Colour standard 0 is pure water and colour standard 10 is pure beetroot extract. So I have my distilled water here and I have my beetroot extract. Okay, and I've got my tubes labelled up here 0, 1, 2, from 0 through to 10. So 0, 2, 4, 6, 8 and 10, right, as is on the table down here. Now you should have filled in the table yourself as part of the pre-practical. If I just scroll this down, there we have it. Okay, so that should be pretty clear as to what we're doing. Of course, for the pure one, for 0, it's going to be 5 versus nothing. So I'm going to get my 5. I'm going to pop that in there. And then, of course, it's just 4, 3, 2, 1 all the way along. So I'm going to go in the next one with 4, and the next one with 3, and the next one with 2, and the next one with 1. Okay, now it's easy in these experiments to get sort of bogged down into the details and not actually look at the tubes. Obviously I've gone 5, 4, 3, 2, 1, 0, so I should have, as I do have here, look, a nice declining sequence. It's often easy to forget. Just look at them sideways and you can see what's going on. Now I'm going to go the other way with the beetroot extract. Very straightforward. So, up to 5, 5, 4, 3, 2, and 1 in the last one. Now you should hopefully be able to see visually a difference in the colours. So we give those all a shake up so they mix up nicely. Give them all a shake up so they mix up nicely. And hopefully you can visually see that we now have a colour spectrum as per the instructions. Okay? Those are going to be what we use to compare our results to. So that there is part 1 of the experiment done. Okay, very straightforward. So let's move on to part number 2, which is here. Okay, so set up a water bath. That's already done. We've got a water bath set up at 30 degrees just here. Now, with a second set of test tubes, so that will be these ones along here. I'm going to use the ones with the corks on them. Second set of test tubes, add 2 cm cubed of the 100% alcohol to a test tube and put the bung in. Label the tube with the alcohol concentration and repeat steps 5 and 6 with the 80, 60, 40 and 20. So I've got my tubes here and underneath this bit of plastic here to prevent evaporation, because of course this is our riskiest bit of the experiment, arguably, is the use of the ethanol. And maybe you could argue the knives, but then you are sick formers and I would expect you to be able to chop things up without cutting yourself. So I just spin this round so you can see what I've got. 100% ethanol, 80% ethanol, 60, 40, 20 and pure water. They're made up in exactly the same ways that I just made these ones here up. Okay, so what I'm going to do, I'm just going to take these bungs out for now. Now I'm going to be pretty fastidious here, and you can debate whether this is necessary or not. We're going to use a different syringe for each of the concentrations, right? Because obviously any residual left in here might alter the percentage of these solutions. Now that probably isn't a massive issue, but you can debate that yourself and think how much of a confounding variable that would be. Because of course spotting control variables is not just at A level about spotting them, it's also about saying how much of an issue they are. Okay, and in this case I am being fastidious. I'm going to use a separate one for each concentration, but arguably there wouldn't necessarily be that much of an impact, certainly when you consider how we're assessing the results. Okay, so here we go. So that's now the 60%. Let's make sure I'm getting these right. There we go. The 40%. Oh, that hasn't worked very well. So always got to check for bubbles in your syringe. 40%. There we go, and compensate accordingly. 20%. 2 mils. Now that there, just as I was saying, look, that's a bubble, right? I'm not overly happy with that. I want to see if I can get a bit better, so I might just tip that up, squeeze the bubble out. I'm going to get a bit of ethanol down the side there. Okay, and measure again, bring that up to 2 mil. Lovely. Okay, so that's nice. And again, you can debate when you've seen the experiment whether the precise quantity of ethanol is a significant confounding variable or if it isn't. Right? Certainly if that's going to make a difference to the overall outcome of the experiment. And then we've got our lovely pure water. Let's go straight in there. Okay, so that's all done now. I'm now going to get those with their bungs in. I'm going to get them warming up. So there we are, pop those in there, pop those in there. And I'm going to get those going on to 30 degrees in my water bath. There we go. Beautiful. Okay, so those are now warming up. We'll leave those as long as it takes 2 ml of water to equilibrate to 30 degrees C. And we'll put the colour standards, our solutions kit to one side for now. And then we're on to step number 10, cutting beetroot slices of 2 millimetres. So, what I'm going to do, set that off to one side, get my little cutting dish out here, got my knife, lovely pre-prepared beetroot cubes, and I find that the easiest way to do this is to use a little ruler. Okay, take your beetroot slices, like so, pop those down. This is where you are going to get murderer hands, I'm afraid, and find yourself a little ruler. There we go. That'll do the job. We lay our ruler down by the beetroot, line them all up, like so. And the quantity I'm aiming for is 10, so carefully cut 10 slices of beetroot, 2 mil in width, and wash them under the tap. So I'm going to get my little sieve ready for that stage, and I'm going to try and cut them 3 at a time. So at 2 mil, lining up the knife, cut those, and 2 mil, cut those. So that's 6. At 2 mil we cut again, making sure of course the knife is straight. Now again, you can debate how much the precision of this really matters. I'm going to do a couple of spares as well, in case I lose them. how accurate you need to be, because what really is important, okay? If we're measuring membranes, then arguably it's not actually the volume, it's the surface area that's important, okay? So that's one argument. Really what's important here is our amount of surface area available. However, you could also make the argument that because we're measuring the amount of pigment that's going to come out of this experiment, then what we're actually measuring as well, right, is we're actually going to be measuring the amount of pigment coming out, so the amount of pigment within the cube to start with is an important factor. Okay, so should have the thermometer over there. Just going to pop those on top there and just give these a rinse off with our water. Now, as I can see there, look, that last bit of water I poured through is running largely clear there. Hopefully you can see that, that's running clear. What we've done that for, of course, is when we cut the beetroot, we're popping cells, okay? And if we're bursting cells, we don't have to do any membrane disruption to get that pigment out. Remember, it's the pigment that's going to give us our result when we compare them to our colour standards. So that's to wash off any excess. Okay, so that's number 10. Number 11, carefully blot the slices of beetroot with a paper towel to remove excess water. Okay, so I'll put my beetroot to one side there. I'm going to pop those onto the paper towel, spread them out, fold the paper towel over, and just blot those. I'm not looking to compress them, because again, of course, what you could do is you could burst some more cells, releasing more pigment and disrupting the result. So what I'm going to do is just blot those nice and gently, peel this back, and hopefully, yep, off they will come there. Okay, now, so that's number 11. Number 12, gently put 2 discs of beetroot into each of the 5 tubes, replace the bungs as soon as possible after doing so, and start the clock. Now, for this one, I can't put in, right, I can't put in 10 slices of beetroot at the same time, so what I'm going to do is what I call Staggered Edition. Really important bit of scientific technique here. So as long as you know the interval between when you put your bits of beetroot in, right, it doesn't really matter. So I might start with my 0% at 0 seconds and put those in, boom, stop clock on, and then when it gets to 20 seconds, then I put my 20% in. 40 seconds, 60 seconds, so on and so forth. And as long as I then stop the experiment at that same staggered sort of idea, because it says here, we're going to leave them for 5 minutes, shaking each time. So as long as I stop them at 5 minutes, 20 seconds, 5 minutes, 40 seconds, 6 minutes, so on and so forth, then they will all have had the equal time, and it will eliminate that practical difficulty of me trying to get 10 slices in at the same time. Okay, so I'm going to start that now. I'm going to take that off. I'll check there at 30. Yeah, that's pretty accurate. I'm going to take that off there. I'll get myself my 2 slices, and in they go. Start the stop clock. Bung on, bung off. I might be able to do this every 10 seconds actually. 7, 8, 9, 10. In they go. Bung on, bung off. 2 slices. 19, 20. In they go. Bung on, bung off. 2 slices. 27, 28, 9, 30. Pop that in. Bung off. 38, 39, 40. Bung in. Bung off. Last 2 slices. And almost missed that one. 50. Okay, brilliant. So the instruction now says leave the tubes in the water for 5 minutes, shake the tubes gently once every minute, and then remove the tubes from the water bath. Immediately pour each solution into a clean test tube. I'm not going to make you wait and watch as I shake these every minute, okay, but obviously I can stagger them. Okay, so we'll now do the magic of TV. Right, 5 minutes will have elapsed now. Okay, we're at 4 minutes now. Just give me 1 minute before I have to start working to explain what I've done here. Just a little bit of a clear down, okay. I've set up a funnel, right, and I've got my filter on the top there. And obviously I did this at 10-second intervals, so I've now set myself up to do 10-second intervals when I take these off. These are my clean tubes, they're labelled 0, 20, 40, 60, 80, 100, and I'm going to take these out in the same order that they went in, starting with the 10% and going up. Now of course what I'm going to need to do then is take these and compare where they are on my colour scale over here. Okay, so I'm just about ready to go. We've got 20 seconds coming along. Got my waste bucket ready for the bits of beetroot. Okay, try and spot any little variables in 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, out we go, through we go, and in the bin. Next, out we go, through we go, and in there. Out we come with the third one, in we go, and back into there. Get rid of that. And next one, in the 60%, in we go, in the bin, next one, in we go, in there, in the bin, and last one, out we come, in we go, in the bin. Okay, broadly kept to time there, which is pretty good, and these are now our results. So if I can take some of these and just pop those back out of the way over here so I can show you what I've done. Here we've got the results. So we can clearly see that if I take the most extreme versions, this side and this side, there's definitely a difference in colour. Okay, so the alcohol hasn't disrupted the membrane very much in water, but it has in 100% ethanol. Now if I go down the row and try and do a comparison to my little colour charts, okay, so it says here, compare each of your solutions with the colour standards you made in part 1. Note which standards has the same colour as each of your solutions. Well, I can go this one here. If I come down my colour standards, well, it's certainly not clear water. Just checking you can see that. It's certainly not clear water. But it also, I would say, is slightly lighter, or maybe even the same-ish colour as that one. So I would give that, I would record in my table a 2 for that one. Next one, coming down, well, it's a bit darker than that. So coming down here, is it about the same? Well, maybe it's a tad darker than that one. So if I come to this one, yeah, I think that's a better match. Okay, so I would give that one a 6. Okay, so that one gets a 6. Next, this one here, well, logically it's got to be darker than the 6. Not much. Yeah, it's probably closest to the 8, I would say. Is it closer to the 10? 10's just a shade darker. So I would say that's an 8. Then this one here, logically, must be somewhere between an 8 and a 10. Maybe a shade darker than that. So I would say that's probably closer to a 10. Okay. And then logic would dictate, if that one's a 10, then these guys here have got to be 10s as well. And I would say that the 80, well, that's a 10. There's something funny going on with 100 over here. That looks arguably slightly lighter. So I think I'm going to give it a 10, but note that actually maybe that is even slightly lighter. And maybe think why it is at 100% alcohol you might end up with a slight decrease in the amount of pigment. Interesting. Okay, now of course that is my experiment. Okay, that's the results that I've got. You might have some obvious criticisms of this method of recording results, okay, and that's good, so you should, because there's a better bit of kit we can do. The problem with this, of course, is subjectivity. This is an entirely subjective way of measuring. I am saying I think it's closest to that colour standard, I think it's closest to this colour. Your eyes might be different to mine. What you saw down the camera could also be very different to what I saw here, right? So that's a subjective thing, and we don't like subjective things. Okay, we like objective things, like quantifiable, enumerable things, as in like things you can put numbers to. Okay, so hopefully you've all come across one of these before. This is a colorimeter, and what a colorimeter does is it actually shines light through a specimen and generates a percentage of the light that makes it to the other side. This thing is called a cuvette. So were I doing this experiment in a slightly better way, I might take a cuvette, fill that with my sample, try and shine blue light through it, because obviously red things absorb blue light. Pop that in the colorimeter. Obviously I will have measured and set this colorimeter to 0. And pop that in there, close the lid, and that would be attached to a measuring device which would give me either a percentage of transmission or a percentage of absorption. So that's certainly an improvement that can be made to this method. Okay, so that is a practical demonstration of how you do required practical 4. Okay, I hope that makes sense, and you can now move on to the post-practical material. Thanks very much.